Hello dear students. In today's video we'll be discussing on physiology of bacteria, or the term which you will be more familiar will be the growth and nutrition of bacteria. So since we have finished the bacterial morphology, or which I would like to call as bacterial anatomy, now we are moving on to physiology, like how we learn in human anatomy and physiology. So physiology means how do the bacteria grow, how they acquire nutrition, how do they divide, replicate, all those things come under physiology. So, bacteria is a prokaryote. So, the division is by binary fission. So, it's a very simple division. In eukaryotes, the method of division is by mitosis. Replication is by mitosis. So, here it is by binary fission. So, it's very simple. You can see a bacterial cell here with the chromosomal DNA with the membrane. So, first, what happened? The division first starts with the DNA. So, there is a replication of DNA. DNA becomes two copies and they separate. And now an invagination comes from the cell membrane and the membrane also divides, the cell also divides and it becomes two daughter cells. So this is it, binary fission. Binary fission means division into two daughter cells. So that is how bacteria divide. It's very simple. Now, how do they grow? How much time they will take for growth? So the division occurs by binary fission, and how much time it takes for a mother bacterial cell to produce two daughter cells. In general, it is 20 minutes. In most of the medically important bacteria, the generation time, what is generation time? Generation time is the time required for a bacterial cell to divide and produce two daughter cells. So one cell to become two, the time required for that is called as the generation time. So it is 20 minutes in most of the medically important bacteria, but there are two exceptions which is very important. That is your mycobacterium. You remember mycobacterium, for which we use the acid-fast staining. So mycobacterium, both tuberculosis and leprae. Leprae causes leprosy. Mycobacterium tuberculosis causes tuberculosis. Mycobacterium tuberculosis require 20 hours. So if it is normally 20 minutes, here it is 20 hours. And Mycobacterium leprae takes 20 days. So that much time is required for a single cell to become two cells. Okay. So why is this important? This time, generation time. So in general, if a bacteria divides into two in 20 minutes, normally a normal bacteria, when you do culture in lab, after 12 to 18 hours, you can get a visible growth in the form of colonies. So in culture plate, you will see bacterial colonies. When they divide and form colonies. A visible colony, you will get a visible colony in culture plate by 12 to 18 hours normally. Whereas in Mycobacterium tuberculosis, since it takes 20 hours, it will take more than 2 weeks. Usually 2 to 8 weeks is required for growing it in cultures. So that is because of the longer generation time. And coming to leprae, you cannot cultivate leprae in any artificial culture medium. We are not able to do culture of leprae in any culture medium. And see, the generation time is 20 days. So if you want to isolate leprae bacilli, you have to inoculate them into animals, not into any culture medium. So just remember the different generation time, 20 minutes for most of the bacteria, 20 hours for Mycobacterium tuberculosis, and it is 20 days for leprae. So again, these generation time is an important MCQ. So please note that. Coming to the most important question, important aspect of bacterial growth and nutrition, it is the bacterial growth curve. So what is meant by this bacterial growth curve? The name indicates curve, so it's some kind of a graph. So suppose you are inoculating bacteria. See, if suppose this is a liquid culture medium, you are inoculating a sample into this culture medium, a bacteria into this, the bacteria will start multiplying in the culture. And if you're going to note the count of bacteria, how many bacteria are present in this culture at different points of time, at different intervals of time, you're noting the bacterial count. And if you're plotting this count of bacteria, so here you're plotting the count and here it is the time. So if you're plotting the count against time in a graph, you get a definitive course. So that if you plot it in graph, you get the growth curve. So when bacteria is inoculated in a culture medium, in a liquid culture medium, it will follow a definitive course of growth. How can you note that growth? You can calculate the number of bacterial cells present in this culture at regular intervals of time, and if you plot it in a graph, you get a bacterial growth curve. So how many cells of bacteria are present in each intervals, like you are noting it after 1 hour or 2 hours, 3 hours, 4 hours, like that. You can get a curve that is called as bacterial growth curve. Now, how you have to see, it's not just simple. If you simply plot a bacteria, normally if the bacteria is replicating every 20 minutes, generation occurs every 20 minutes, you have to get a rising curve, right? But it's not like that. The curve is not like that. You can see initially it is a very lagging phase. There is no increase in number. Then the numbers are increasing exponentially. Then it is remaining stationary, and finally it is coming to a decline phase. So these things happen because the nutrition available in the medium is not static. It will get depleted. Bacteria will produce its own toxic products. Bacteria will release autolytic enzymes. So all these factors will come into play and determine how the growth occurs in this culture medium. So that is why you get this kind of curve. So we'll see what happens in each phase. So the first phase is lag phase. So remember, you can remember when we are going to do a new thing, initially every one of us will be having a lag phase. No, we get some time to have a flow, or to start with you need a little bit time. So that time bacteria also requires, that is called a lag phase. So in this time this bacteria is not simply sitting, it's making all the preparations. Like what we do, we do some mental preparation, right, to start a new thing. Like that the bacteria also do some preparation. What they do is they will collect all the enzymes, they will produce all the enzymes, all the nutrients that it require, metabolites, everything it will collect in this time so that it is ready to start replication. So the bacterial cell will accumulate metabolites, produce enzymes, accumulate nutrients, and it is ready to start dividing. So by that time what happens? Bacteria will increase in size. So lag phase is bacteria is simply sitting and preparing. So generally it will increase in size, it won't divide. Okay, so lag phase is the time where there is period between inoculation and start of multiplication. So the time between inoculation and the start of multiplication is called as the lag phase. So in this phase there is only increase in the size of the cells. There is no increase in number of the cells. Okay, once the bacteria starts dividing, then what happens? It will be in a logarithmic phase or an exponential phase. So there will be increase in number, exponential increase in number with time. So this is the time where the cell division is occurring very smoothly and the bacterial count will be increasing exponentially. So you get a straight curve or a straight line graph. That is the exponential phase. So maximum number of bacterial count you will get at the end of exponential phase. So what happened afterwards? After exponential phase, you know what happened? Bacteria is using all the things, it is replicating, using all the nutrients. So nutrients will start getting depleted and it will start accumulating the toxic products. So now what happens is it will start dying. Some bacteria will die. Some others will try to replicate. So there is a balance between the dying cells and the newly formed cells. So the number of bacteria newly formed is equal to the number of bacteria which is dying. So there is a balance between these two. So in effect, what happened when you plot the number of viable cells? I'm telling viable cells. That means we are noting only the live cells. You get a stationary state. The viable count is not increasing. Why it is not increasing? The number of cells dying is equal to the number of new cells produced. So that is called the stationary phase. So number of viable cells will remain stationary. So one more thing you note, in exponential phase, now bacteria is very happy. It is dividing with lot of nutrients. It is increasing in number. So that is the phase when bacteria look most beautiful in microscopic smears, in staining. So when you do staining, all the bacteria will be uniformly stained, uniform size. All that beautiful features of bacteria will be available in the exponential phase. When it comes to stationary phase, now it has started phasing. depletion of nutrients, and it has started dying. No, some bacteria starts dying. So in that phase, bacterial staining will be very poor. It will be irregular staining in the stationary phase. And one more thing, you can imagine what is the form of bacteria that is produced when there is depletion of nutrients. Stationary phase is the phase when bacteria produce spore. How you can remember S and S, right? Stationary bacteria produce spore. Finally, what happened? It's a phase of decline or death phase. Now there is no more nutrients available, lot of toxic products accumulated, bacteria is releasing its own autolytic enzymes, so no more replication happens, only death of bacteria happens. So the viable count will just fall down. Decline phase. So after stationary phase, there is a decline phase. So that is what happened in the phase of decline. And one form you can remember, you remember what you learned in morphology, the involution form. Aberrant forms of bacteria, involution forms, you can see the involution forms in phase of decline. So that is the bacterial growth curve. So it's very important, please learn to draw this lag phase, log phase, stationary phase and decline phase. So please remember some points which are very important. I think you'll be able to answer by now what is the maximum phase at which you get a maximum size of bacteria. That is a lag phase. You can remember like a lockdown phase. At the end of lockdown phase, all of us have gained weight. Similarly, at the end of lag phase, it's a lockdown phase for bacteria where it's simply accumulating all the things. So it will increase in size. So at the end of lag phase, you get maximum size. And when will you get the maximum number? It is at the end of exponential phase. Maximum number at the end of exponential phase. And when will you get uniform size and uniform staining of bacteria? I told you it is happening in the log phase or exponential phase when bacteria is happily dividing. So everything will be beautiful in the exponential phase. What happened in the stationary phase? Stationary phase, bacteria is deprived of nutrients, so it will form spores and also the staining will be irregular. And finally, in the decline phase, you will get the involution form. So maximum size at the end of lag phase, maximum number at the end of log phase, and uniform size and staining is obtained in log phase. Spore formation is seen in stationary phase, S4S, and involution forms are seen in the, involution means it's aberrant forms, swollen aberrant forms. So that is seen in the death phase. So these are the different points. All these can be asked as MCQs, and bacterial growth curve is an important question, 5-mark question, which is asked for the exams. Okay. Now, one more thing you can see, though this is not important in the exam point, you should know what are the factors which affect bacterial growth, like moisture. Like any other living being, it requires water. 80% of the bacterial cell is composed of water, so it definitely requires moisture. Oxygen, carbon dioxide, temperature is important, adequate temperature, adequate pH, light, and the things which can affect bacterial growth is osmotic changes, mechanical stress and all. Regarding oxygen, some things you should note. Sometimes you can be asked as a question, aerobes, anaerobes and microaerophilic organisms. Based on the oxygen requirement of microorganisms, they are classified as aerobes and anaerobes. So what is meant by aerobes? Aerobic means they require oxygen. Anaerobic means not aerobic. They don't require oxygen. So aerobes can be classified as obligate aerobes. Obligate means strict aerobes. They definitely need oxygen. Example is Pseudomonas. Pseudomonas is a bacteria which cannot grow without oxygen. So it's called an obligate aerobe. Now, what is meant by facultative anaerobe means they are normally aerobes, but even without oxygen, they can survive. Facultatively, they can act as anaerobes. Such bacteria, example is E. coli. E. coli is an aerobe, but it can even survive without oxygen, so it is called as facultative anaerobes. And what is meant by anaerobes? I told you no oxygen. And if it is an obligate anaerobe or a strict anaerobe, even minute traces of oxygen is harmful to this bacteria. Example is Clostridium, Clostridium tetani, Clostridium perfringens, all those tetanus causing Clostridium tetani, they doesn't require oxygen at all. If oxygen is present, they cannot grow, they cannot survive. And some bacteria, they need only very less amount of oxygen. That's why it's called microaerophilic. Aerophilic, though it is aerophilic, it needs it only in very micro quantity. An example for a microaerophilic bacteria is Helicobacter. Okay, so aerobes and microaerophilic. Please remember the examples of an obligate aerobe, Pseudomonas, facultative anaerobe, E. coli, obligate anaerobe, Clostridium, and a microaerophilic organism, Helicobacter. And regarding carbon dioxide requirement, most of them need only very small quantity of carbon dioxide, but there are exceptions. Some of them have a special affinity for carbon dioxide that is called as capnophilic. Philic means it loves carbon dioxide. So example is Brucella. Brucella requires 5 to 10% carbon dioxide. Okay, that's one exception. And regarding temperature, we all know temperature. Most of the bacteria love human body temperature. They survive easily in the human body temperature. So what is the human body temperature? 37 degrees Celsius, right? So most bacteria like the temperature 37. So between 25 and 40, if they are surviving, you call it as mesophiles. If they survive at very low temperature, you call psychrophiles, low temperature. Those are actually saprophytes, soil saprophytes, water saprophytes, which are normally seen in the environment. Such bacteria can survive at lower temperatures. And what is meant by thermophile? Thermophilic means temperature loving. It loves high temperature, 55 and 80 degrees Celsius, even higher than that. Example for such a thermophilic bacteria is Bacillus Stearothermophilus. Please note this bacteria name because it will come under sterilization when you learn about autoclave. Stearothermophilus. The name itself indicates it is temperature loving. Stearothermophilus. It requires high temperature. Not requires, it can live in very high temperatures. So those are the different temperature requirement wise. You can classify bacteria as mesophiles, psychrophiles, and thermophiles. So that's about the growth and nutrition of bacteria. And I'm repeating, bacterial growth curve is the most important question from this part. Please learn to draw this and what all happens in different phases: lag, log, stationary, and decline phase. Thank you.